Hello, my name is Carly Rundle, and today I will be presenting research conducted at the University of Illinois in collaboration with DuPont Animal Nutrition, investigating the effects of including Buttiauxella phytase to diets based on corn, soybean meal, and canola meal on the growth performance of weanling pigs. Phytate is the main form of phosphorus found in plant feed ingredients, including seeds, nuts, legumes, and grains, with about 50 to 60% of phosphorus bound to phytate. This phytate can form complexes with minerals including zinc, copper, manganese, calcium, and iron, and therefore reduces the digestibility and absorption of many minerals. As phosphorus is a required nutrient for bone formation, acid-base balance, and general metabolic function, it is crucial that pigs obtain optimum levels of phosphorus through the diet. Phytase is an enzyme that catalyzes the release of phytate complexes. However, the intrinsic concentration of phytase is very low in corn and legumes, common ingredients in diets fed to pigs in the United States. There is also some phytase expressed in the gastrointestinal tract of pigs, which results in additional cleaving of phytate, yielding available phosphorus and calcium for absorption by the pig. However, this is not enough for optimal utilization of feed ingredients. As a result, exogenous microbial phytase produced on a large scale through cellulosic biomass fermentation is added to diets fed to pigs to maximize digestion and absorption of phosphorus in order to reduce feed costs associated with including high levels of inorganic sources of phosphorus. Additionally, phasing out of high inorganic mineral concentration reduces mineral deposition in manure, which reduces the impact of livestock production on the environment. This increase in digestion and absorption of phosphorus has been shown to improve the growth performance of pigs, including increases in average daily gain and gain to feed, and it has also been shown to improve bone strength and bone density, resulting in an improvement in overall health of the animal. Including phytase in diets fed to pigs may then result in a reduction in feed cost, as improvements to growth efficiency are made and organic sources of phosphorus, including corn and legumes, are utilized more efficiently, enabling producers to reduce amounts of inorganic calcium and phosphorus in the diet formulation. Previous research conducted in the Stein Monogastric Nutrition Laboratory investigated the effects of phytase inclusion on the apparent total tract digestibility and fecal excretion of calcium and phosphorus by weanling pigs. It was observed that inclusion of 250, 500, or 1,000 phytase units, or FTU, per kilogram of Buttiauxella phytase increased the apparent total tract digestibility of both calcium and phosphorus when fed to weanling pigs, compared with a negative control diet that was formulated without a source of inorganic phosphorus. Apparent total tract digestibility of calcium and phosphorus was also quadratically increased as phytase inclusion in the diet increased, with the greatest digestibility observed in the 1,000 FTU phytase diet. In addition, this phytase inclusion improved the absorption and utilization of feed ingredients, evidenced by reducing the fecal excretion of calcium and phosphorus, leading to the conclusion that Buttiauxella phytase would reduce mineral deposition in manure. Therefore, the next step in testing this source of phytase was to ensure that this product not only increased digestibility of calcium and phosphorus and reduced fecal excretion of minerals, but also increased growth performance indices, including average daily gain and gain to feed ratio. We speculated that due to the increase in digestibility of minerals, we may reduce the inclusion level of inorganic sources of calcium and phosphorus, including limestone and monocalcium phosphate, in the diet, and still obtain ideal growth performance compared with a conventionally formulated nursery diet. Inclusion of phytase in diets fed to growing pigs improves the digestibility of calcium and phosphorus, and consequently may improve growth performance of pigs fed diets with reduced nutrient composition. Therefore, it was the hypothesis of this study that Buttiauxella phytase from T. reesi would ameliorate the effects of reducing concentrations of nutrients in the diet on growth performance compared with a conventionally formulated diet when fed to weanling pigs. There were 5 experimental diets in total. A positive control or PC diet based on corn, canola meal, and soybean meal was formulated with monocalcium phosphate and limestone, to contain 0.33% digestible phosphorus and 0.60% total calcium. The negative control, or NC diet, was then based on corn, canola meal, and soybean meal, and formulated to contain 0.18% digestible phosphorus and 0.45% total calcium, with limestone and monocalcium phosphate included in the diet in reduced concentrations. Standardized ileal digestible amino acids were decreased by between 0.01 and 0.02% in the diet, and digestible energy concentration was reduced by around 43 kilocalories per kilogram between the positive control and the negative control. 3 additional diets were formulated by adding either 250, 500, or 1,000 phytase units per kilogram, or FTU, of Buttiauxella phytase obtained from T. reesi to the negative control diet. This experiment was conducted for 21 days. Pigs were weighed at the beginning and conclusion of the experiment. Daily feed allotments were recorded, and feeders were weighed at the end of the experimental period to calculate feed intake. Average daily gain, average daily feed intake, and gain-to-feed ratio were calculated for each treatment. Data were analyzed using contrast statements to compare the positive control and negative control diets, positive control and phytase diets, and to determine the linear and quadratic effects of phytase inclusion. Now we will move on to the results of this experiment. Setting up the graphs, we have phytase inclusion on the x-axis, with PC indicating the positive control diet, NC as the negative control diet, and 250, 500, or 1,000 indicating the inclusion level of Buttiauxella phytase in the diet in FTU, or phytase units per kilogram. First off, looking at final body weight, we did not observe an effect of phytase inclusion on the final body weight of the pigs compared with the pigs fed the control diets. While there does appear to be a slight numerical reduction in the final body weight of the pigs fed the negative control diet, this difference was not significant compared with the phytase diets or with the positive control diet. Next, looking at the average daily feed intake of the pigs, there was no difference between the pigs fed the positive and negative control treatments. Average daily feed intake linearly increased as phytase was added to the diet, with the greatest feed intake by the pigs fed the 1,000 FTU phytase diet. The pigs fed the negative control diet had reduced average daily gain compared with the pigs fed the positive control diet. However, this reduction in gain in the negative control was ameliorated when phytase was included at 250, 500, or 1,000 FTU per kilogram. Additionally, average daily gain quadratically increased as phytase increased in the diet, with the greatest gain observed in the pigs fed the 1,000 FTU phytase diet. Gain to feed was reduced in the negative control treatment group compared with the positive control group. Addition of phytase resulted in a quadratic increase in the gain to feed ratio, with the greatest gain to feed observed in the 500 FTU treatment group. Overall, these data suggest that inclusion of phytase to diets with reduced nutrient composition, such as the negative control, will increase growth performance indices so that there is no difference between the phytase supplemented diets and the positive control diet. From this work, we conclude that Buttiauxella phytase may be included in diets with reduced nutrient composition and energy for weanling pigs without compromising pig growth performance, compared with a conventionally formulated nursery diet. The data from this study indicate that producers may formulate diets slightly lower in inorganic calcium and phosphorus, reduce feed cost, and still maintain the growth performance of their pigs. I would like to thank DuPont Animal Nutrition for their financial support of this research. And with that, I would like to thank you for listening to my presentation. My email is rundle2@illinois.edu, and if you would like to know more about the research conducted in the Stein Monogastric Nutrition Laboratory, feel free to visit our website at nutrition.ansi.illinois.edu.